Alright, this is the third part of a four-part lecture series to wrap up metabolism. We're going to move on to respiration, first discussing the citric acid cycle. The goals here are to consider what happens after stage 1 and 2 of glycolysis in respiratory organisms that are doing respiration. I want you to appreciate that all respiratory organisms are not necessarily using aerobic respiration. Remember, anaerobic respiration is very important in the microbial world. I'm going to review the citric acid cycle with you, and I want you to also appreciate how the citric acid cycle is associated with biosynthesis. The citric acid cycle is the same thing as the Krebs cycle. Okay? Oops, one too many B's there. So I might use that term, those terms, interchangeably. Alright, many microbes exist that are able to use respiration, whether it's anaerobic or aerobic respiration. They have the enzymes necessary and the genes for the proteins important in the electron transport chain that they need to have. Fermentation does produce a lot of ATP. Well, not a lot, but it produces enough ATP to make a living. And we just talked about fermentation previously. However, fermentation is not actually that efficient as compared to respiration, because the electron donating molecule, for example glucose in the examples we discussed previously, are not fully oxidized to CO2. So some electrons remain with those electron donating molecules, some chemical or potential energy remains unused or is wasted. Whereas respiratory processes are much more efficient at removing all of the available energy, all of the available electrons from the electron donating molecule. Microbes that are capable of conducting respiration are able to use a more powerful exogenous, okay, as opposed to endogenous. Exogenous means from the outside of the metabolism you're considering. A more powerful exogenous terminal electron acceptor, like oxygen or nitrate. And in doing so, they're able to fully oxidize the organic electron donor, such as glucose, to carbon dioxide. In most of these respiratory microbes, stages 1 and 2 of glycolysis will happen. They will run, and then the pyruvate that is generated will continue getting oxidized in the Krebs cycle. So in most of these organisms, actually let me correct myself, in all respiratory organisms, when they're doing respiration, they are actually deriving the majority of their ATP from oxidative phosphorylation, not from substrate level phosphorylation, but from oxidative phosphorylation. So let's think broadly about respiration, and let's begin with the model of aerobic respiration, because you should be very familiar with that process. So this particular figure is not a cartoon of a bacterium. Instead, it's a cartoon of a mitochondrion. Okay, this is a mitochondria. However, since mitochondria used to be bacteria, remember the endosymbiont theory that states that in the evolution of eukaryotic cells, bacteria were acquired by other bacteria-like organisms, and those phagocytosed organisms actually persisted and eventually became cell structures known as mitochondria. Similarly, that is how we think that the chloroplasts evolved. Anyway, so back to respiration. So let's take a closer look at this figure. This cartoon shows how in a respiratory organism, glycolysis here will yield pyruvate. And so pyruvate's not actually written in here, but pyruvate is coming out of glycolysis. There are 2 pyruvates coming out for every 1 glucose that went in. And what will happen to the pyruvate is it will react with acetyl-CoA in something called the preparatory reactions. A little CO2 will be kicked out, and some reduced NAD in the form of NADH will be kicked out from those preparatory reactions. And what will happen then is the result will enter the Krebs cycle, or the TCA cycle, shown here in this circle. Okay, so what will happen in the Krebs cycle is all of that pyruvate will be fully oxidized to CO2. So coming out of the Krebs cycle is CO2. 6 CO2 are going to come out of the preparatory reactions and the Krebs cycle process. And that will account for all 6 carbons that were originally in the glucose. So this represents a full oxidation. So those CO2 molecules will diffuse out. The electrons that were stripped out of pyruvate have all made their way onto NADH and FADH2. And in respiratory organisms, what they are going to predominantly do is go off to the electron transport chain. And we're going to talk about the electron transport chain in the next part of this lecture series. But it is at the electron transport chain where the electrons will finally meet their terminal electron acceptor, oxygen in the case of aerobic respiration, nitrate in the case of some forms of anaerobic respiration, or sulfate, or certain iron forms, etc., etc. Anyway, the electrons will finally meet the terminal electron acceptor, and it will be reduced, okay, to, in this case, water. Okay, and as the electron transport chain does its work, it generates a proton motive force. The proton motive force energizes ATP synthase, and the ATP synthase enables the production of a significantly larger number of ATP than what we saw due to simple substrate level phosphorylation and glycolysis itself. So that number 36 is not entirely accurate. It's more sort of in the range of 30 to 38, maybe 28 to 38, depending on the organism and the conditions involved. Let's consider the Krebs cycle in more detail. Alright, so here is the citric acid cycle, also known as the Krebs cycle. Up here we've got pyruvate. Each pyruvate has 3 carbons associated with it, and in here we have the preparatory reactions. So pyruvate from the end of stage 2 of glycolysis is decarboxylated and reacted with coenzyme A, and this will result in the production of acetyl-CoA, right, which is shown here. And it is actually acetyl-CoA which will enter the Krebs cycle. During those preparatory reactions, one oxidized NAD plus is reduced to NADH, and that's going to go off to the electron transport chain, and 1 CO2 is produced. So remember that pyruvate is a 3-carbon molecule, so we've now accounted for one of those 3 carbons, but we have 2 left. They are ultimately going to form CO2 during the cycling of the Krebs cycle. Acetyl-CoA will combine with oxaloacetate, forming citric acid, or citrate. Citrate is converted to aconitate, which is converted to isocitrate. These are essentially rearrangement reactions, what are happening in here, okay, not redox reactions. Isocitrate is then oxidized to form alpha-ketoglutarate. So there's a redox reaction. Okay, and where do we see? We see our good friend NAD plus, or in some organisms NADP plus, and it is getting reduced to its reduced form, and that is going to go off to the electron transport chain. We also see the loss of a CO2, so now we've accounted for 2 of the 3 carbons originally in pyruvate. The alpha-ketoglutarate is further oxidized, transferring electrons to NAD+, and coenzyme A acting in tandem produce NADH. So here we have another reaction, another redox reaction. We have another reduced form of NAD, the NADH formed. It's going to go off to the electron transport chain, and we have accounted for the 3rd and final carbon originally present in pyruvate, so all of the carbons have now been accounted for. The alpha-ketoglutarate was converted to succinyl-CoA, and succinyl-CoA will react with guanosine diphosphate and a phosphate group, yielding guanosine triphosphate, a high-energy molecule that is able to participate in energy coupling reactions and thereby drive many anabolic processes in the cell. Coenzyme A is released, and it is then able to participate in other cellular reactions, for example, circling back around and participating in the preparatory reactions that affect pyruvate, allowing it to enter the Krebs cycle in the first place. So the production of GTP is a substrate level phosphorylation reaction at this point. It yields succinate, and the succinate will be oxidized to form fumarate, so that's a redox reaction. Okay, and the electrons are going to be used to reduce FAD to FADH2, and it's going to go off to, you should have guessed this by now, the electron transfer chain. The fumarate is then rearranged to malate, and the malate participates in another redox reaction where electrons present in malate are used to reduce NAD+, making NADH, and that's going to go off to, you guessed it, the electron transport chain. That will yield oxaloacetate again, allowing the Krebs cycle to continue turning. Okay, so remember, for every original glucose, 2 pyruvate are formed, thus 2 preparatory reactions and 2 turns of the Krebs cycle have to occur in order to account for all of the carbon originally present in glucose. Overall, there will be a net yield of 3 NADH from the preparatory and cycle reactions, 1 FADH2, and 1 NADPH. That represents a lot of reduced coenzymes, especially when you double that to account for all the carbons in glucose. These will be oxidized by the electron transport chain to harness their energy and apply it to the production of the proton motive force. And with that, let's move to the next slide. When you're introduced to the citric acid cycle or the Krebs cycle or the tricarboxylic acid cycle, all those names apply, we really focus on its role in catabolism, but it's also important to understand that many of the metabolites that are present in the Krebs cycle are very important precursors in biosynthetic processes. For example, several amino acids are made using alpha-ketoglutarate and oxaloacetate. So these can be titrated out of the Krebs cycle and fed into the synthesis of amino acids. Oxaloacetate is used to make the high-energy molecule phosphoenol pyruvate, which is a precursor to glucose, which many cells will synthesize. Acetate is bound to acetyl-CoA and released in the preparatory reactions. Acetate, which is part of acetyl-CoA and is then released during the preparatory reactions, feeds into fatty acid biosynthesis, so that's really important in membranes. And succinyl-CoA is an important part of an important precursor molecule in the synthesis of cytochromes, chlorophyll, and other tetrapyrrole compounds used by a variety of cells for several different purposes. We'll think about the cytochromes as we think about electron transport chains in the next and last part of this 4-part lecture on metabolism. And this concludes this particular part, part C of part 3, Lecture 5.